Hi students, I wanted to make a short video just with a review of the enzymes that are part of the DNA replication, and for that end I make a little blackboard here on my iPad to show you a little brief summary of the enzymes that you studied during class. Okay, so let's go back here and let's just briefly review the DNA duplication. So during the demonstration in class with the little blue grommet and the cord, we studied three important enzymes. Those were helicase, topoisomerase, and the single strand binding molecules. Okay, so in this drawing, if I were to draw those three, we will have here a helicase that I'm gonna draw like a small triangle, right? And then I will have the single here, the single stranded binding molecules, or SSB, holding apart the two strands of DNA. And over here somewhere, I will have the topoisomerase that is uncoiling the supercoiled DNA because of the movement of the helicase. So coating the phosphodiester bond, that is a covalent bond, the sugar phosphate backbone of DNA, and reattaching it to keep the helicase working smoothly. If you don't remember this, we have posted already the video of the demonstration in the YouTube channel. Okay, so we're at this point. We're going to assume that this DNA goes from the 3' end to the 5' end here on bottom. And because the strands are complementary, remember the word complementary, what that means? That means that the opposite strand goes from the 5' to the 3' direction, okay? So what we know at this point is that we call it 5' and 3' ends depending on what is the attached, right? The 3' end is because in carbon number 3, there is a hydroxyl group for DNA, right? And for 5' we have in the 5th carbon of the molecule the phosphate group. So if you remember, there was like a phosphate group right there. Okay, so remember how to enumerate your carbons, right? So to know if it is DNA or RNA, in the second carbon, you will have to look for a hydrogen or hydroxyl group. But in the third carbon, there's always hydrogen. And the reaction, it is dehydration reaction between the hydroxyl group and the phosphate group that actually makes the phosphodiester bonds on the sugar phosphate backbone of DNA. Okay, so we're at this point and we want to copy this strand. So the first enzyme that is involved in duplication is a really pretty enzyme that I'm going to draw in yellow that is going to be called the primase. Okay, these primers take something that is called the primer. That's what the word comes from. So the primer are like 10 to 12 nucleotides of RNA, and this is important, this is RNA, that can attach directly to the template of DNA. So I'm going to touch it here, right? So G goes with C, right? And A, usually in DNA, will go with T, except that in this case, it won't directly go with T. It will go with uracil because we're talking about DNA, right? And then here, it will attach a U. So this is a primer, right? I draw it here really, really small, but in reality, it's like 10 to 12 nucleotides that should attach there, right? Now, important for the primase and any other enzyme involved with nucleotide attaching is that all of them are always going to move from the 5' to the 3' direction, right? So this way. That means that your leading strand is always the one that goes from the 3' to the 5' direction. And the reason why it's called leading is because it's really smooth, like a zipper, right? That goes right straight through. Okay, so we have a primer that was put by the primase, that is an RNA enzyme. And the next beautiful enzyme that is going to appear here is the DNA polymerase 3. So let's draw that in another color, like blue, right? So DNA polymerase 3. 1, 2, 3. Sorry for the writing, but this is a DNA enzyme, right? Once the primer has been put there, we have a starting point, okay? So now DNA polymerase 3 can work really, really nicely in a very simple fashion here, A, right? And it will continue on and on and on with the same direction that the helicase goes. So if you are confused about the direction, just look at the direction at which helicase is going, and that should be always the leading strand, okay? So we have the primer, we have now pieces of DNA that continuously go, and this looks a little weird, right? Because now you have like a yellow RNA piece, and then you have the blue DNA piece. And if you wanted to duplicate DNA, that's not exactly what you want to do, right? So for that, we have 2 more enzymes that come to the rescue. The next enzyme, let's write in another color, let's say not yellow, but green here, right? The next enzyme that is going to make its appearance is going to be DNA polymerase 1. And of course, this is a molecule of DNA, and this is going to be responsible for, instead of keeping these primers with the RNA, switching them to be our beautiful DNA. So in reality, what it will do, right, is remove the hydrogen on carbon 2 for the hydroxyl group of RNA, converting it into RNA, and wherever there's a uracil, is gonna switch it to T, right? So if I were a DNA polymerase 1 in my iPad, what I would do is I pretty much will erase this template, which is not true, it's just a chemical reaction, right? But I will erase this template. And the next thing I will do is I will put the correct, right, nucleotide here. So T, T. The next thing you can notice in here is that I still, even though I have now full DNA, right, it's not quite great because we have like a green part and a blue part. So the next enzyme involved with duplicating, and I'm gonna put it in purple here, is ligase. And in Spanish or in Latin, liga means to link, right? Li, to link. So what this means is that this enzyme is responsible for linking these 2 pieces that seem unattached, right? And actually linking the whole thing to make a really neat and a smooth phosphodiester series of bonds, okay? So that's it for the leading strand. Let's go to the lagging strand, right? Okay, so I have again my DNA here very, very clean, right? And the lagging strand is slightly more difficult. And the reason why it's slightly more difficult is because this goes from 5' to 3', right? And then when I wanted to move any of the enzymes involved in duplication, I would like to move from 5' to 3'. And I cannot do that here because this is the anti-parallel nature of DNA, right? Is that I will have to move it smoothly here, I will have to move from 3' to 5'. And enzymes, they just don't know how to do this. And in class, we talk about how some things in biology are more difficult than they need to be. And this is one of those things where why isn't an enzyme capable of moving in the extra direction, right? Why didn't we evolve to have that enzyme that moves in the antiparallel fashion? And we don't know. It just hasn't mutated. Maybe at some point it mutates and it takes over DNA replication, right? But at this point, that enzyme doesn't exist. And all our enzymes are moving from 5' to 3'. And as you can see here, they will be forced to move in the difficult direction. So if we have primase, right? So our original primase that puts primers, let's say it will put the primers here, right? A of RNA, A, G. This is RNA, remember, primase. And if I wanted to continue in a really cute fashion, I could go this way, right? Except that the big question here is, big question, what happens in this part? We cannot have DNA without a part that is not being read. That is not good, right? So the solution for that is that our beautiful primase is actually going to put another, and I'm gonna delete here, I'm gonna delete this. Is going to put another primer right before a G here, and it's going to leave this space open right here. This space keeps open. And this is where the DNA polymerase can work, right? So our DNA polymerase was a blue one. And this DNA polymerase can move here, T, C, and T, can move in the right direction, the 5' to 3' direction, right? So now you see that it is a sandwich of DNA of the sequences that DNA polymerase 3 put between here, between, I want to point out, here's the one primer, here's another primer, and this piece is sandwich in between those two. Okay, so because it is sandwich, right? We call this sandwich. Famous name, Okazaki fragment. And this happens on and on and on in the lagging strand. The next two steps are easy, are exact same steps that happen in the leading strand. Having a DNA polymerase, so the green one that we had before, replacing this for DNA. And there's no RNA anymore. And the next beautiful step is our ligase linking all these fragments over and over to make a new phosphodiester bond. So this is how these four enzymes, the ligase, the DNA polymerase 3, and the DNA polymerase 1, and the RNA primase form two copies, right, of DNA and the semi-conservative DNA replication. I hope you enjoy it and understand this, this video. Bye.